And
All right, welcome back to the afternoon session. We're going to sort of switch gears from exploratory data analysis, where we're sort of looking at the data in an unsupervised manner, to more of a targeted analysis where we have an outcome of interest. And now we're actually interested in identifying individual variables that are highly associated with the response. And which can generalize to new data sets that's currently unseen with the data set at hand. And so this session is on biomarker discovery and evaluation techniques. I want to start off by acknowledging the land that we gather. This is a traditional, ancestral, and unceded territory of the Musqueam people, and we are grateful that we are on it and can use it. This is the copyright disclaimer. And I have in the chat, if anyone is currently on Sockeye, the instructions to open up the RStudio session that I am currently in right now. The way you do it is you copy the biomarker discovery folder from the project space into your own user space. And then you should be able to modify the run RPy Studio shell script, which is essentially the job script, and you only need to modify the allocation code, which is st-strv-1, and then add your email as well there. And then that once you're able to run the job file, you'll get a connection script, which you should open a new terminal window and input the SSH details, and then log into your RStudio server. And once there, you specify the lib paths to where the RStudio file path is for the R packages, sorry. And essentially it should be the same path as your user area for the biomarker discovery folder. And I think I should have it here as well. This should look something like this, except you replace this bit with libs _ 4.1.0, because that's the folder that's on Sockeye. And once you have that, then you have all the packages available for today's presentation, and you should be able to knit the document or run all the commands. And for the purposes of today's talk, I'm using a built-in plugin called Infinite Moon Reader, which just generates the slides based on this R Markdown, so I can navigate using this as well. So let's move on to the learning outcomes. So the 4 learning outcomes for today include just knowing a lot of definitions around biomarkers and evaluation techniques. How do you determine performances of biomarkers and panels of biomarkers? And so it's very important to know what those are, and especially how to use them in practice. And we're going to use both simulated and breast cancer data set to identify biomarkers, and that's learning outcome 2. Learning outcome 3 is knowing how to discriminate between the wrong and the right way of evaluating whether or not a biomarker will generalize to new populations, right? So oftentimes in the literature, when you have a given data set and you identify a biomarker, what is the correct strategy of identifying its generalizable error? And lastly, I just want the audience today to get a feel for how easy it can be to really cherry pick the data and identify markers that look amazing at initial glance, but are completely the outcome of hacking away at the data set. So hopefully I am able to convey these messages to you during the next 2 hours. Let's start off with a definition. I'll keep monitoring the chat as we go along. And so the first definition is a biomarker. So what is this? So this definition is coming from the FDA NIH group called the BEST Biomarkers, Endpoints, and Other Tools resource from 2016. And essentially is defined as a characteristic that is measured as an indicator of normal biological processes, pathogen processes, or biological responses to an exposure or intervention, including therapeutic interventions. So you can see it's quite a mouthful, and there's a lot of things it's trying to cover under its umbrella. And you can see that it's being sub-stratified into these various categories. So the different types of biomarkers, and I'm listing various types here. So diagnostic, monitoring, prognostics. And biomarkers can come in various shapes and forms, right? So there are mostly like quantitative measurements, so like molecular histology, radiologically, and they're not necessarily like subjective measurements, like how you feel is not a biomarker, right? Although like how you feel, and if it translates into one of these quantifiable measurements, then yes, that's a biomarker. So the next slide is looking at types of biomarkers. So here I've sort of stratified them across different types. So here this is should be type. So this is a cool thing about our Markdown. I can fix my mistakes as I speak. So here we have 4 types of biomarkers: diagnostic, monitoring, response, and prognostic. And these are individual like single molecule measurements for a given type of biomarker. So for kidney disease, we have GFR, right? Glomerular filtration rate. Cystic fibrosis, you can measure sweat chloride. And these are more of like diagnosing the disease, whereas these other markers are specifically for different purposes, right? So PSA for monitoring. CRP is a very generic biomarker, and it can be used to measure a response to tobacco use. And everyone probably may have heard of the BRCA1 prognostic mutations for determining likelihood of getting breast cancer and recurrence of breast cancer. Any questions so far? Feel free to stop me at any time if you have questions about these. Okay. So I've given you some examples of biomarkers, right? And so in common practice in the clinic, there's a very large preponderance of like single molecule biomarkers, and this really revolves around this concept of this magic bullet that will solve everything, right? So do they really exist, right? Are these like, is one molecule enough to stratify patients or give us the answers we're looking for, right? So here's an example of a biomarker of heart failure. And so this is called N-terminal probrain natriuretic peptide, NT-proBNP. And so essentially it's a marker of like heart stress. And the more stress to your heart is, the more of this molecule you're going to find in the blood, right? So a given cutoff of this molecule at 300 nanograms per liter is an excellent rule out test for heart failure. So if your levels are below this, you probably don't have heart failure, and the sensitivity is quite high, right? So it does a pretty good job of doing that. However, it misses a lot of people that have heart failure at the same time, right? So the specificity just means a lot of people with heart failure that have levels that are quite low as well, right? So this is a meta-analysis showing a lot of these studies that conducted essentially these studies where they measured NT-proBNP in healthy and heart failure patients. And you can see it's very good at ruling out like healthy controls, but it's not very good at picking up heart failure patients that have very low levels of this molecule. So this just speaks to the heterogeneity of the disease. It's not heart failure is not just specific to the heart, right? There's a lot of comorbidities that go along with heart failure, like diabetes, kidney disease, and so on. So then how can we do better? And so this is where the motivation of a multi-marker approach comes into play. And if you're following along on our studio, you can actually click to the next slides with your cursor and it'll navigate for you. And so this diagram, if you bear with me for a minute, if you focus just on the x-axis, you see a single molecule. It's termed gene 1. And you can see the individual histograms of the two populations. You can think of them as heart failure and controls. And you can see there's quite a bit of overlap in the values that occur in the two groups, right? However, as you start adding additional biomarkers, or these additional genes in this case, you can see that the plane of separation, instead of being vertical along the x-axis, is actually made up of a 2D space that is trying to separate these two groups, right? So you have a much better separation between these two individuals when you look at this higher dimensional space that these individuals actually live in, if you look at two markers versus one. And so this is sort of the motivation around why a multi-marker approach or a panel of molecules can give you a much better performance in sort of stratifying patients or making some sort of predictions on a group of individuals. And so at the bottom, there's this equation, right? So you have a biomarker score for a subject, and this is essentially a weighted combination. This is, again, an example. You can get very complicated equations as well, where each individual gene is weighted by a coefficient. And so those coefficients determine contributions of the individual biomarkers to the biomarker score. And that's essentially the idea around developing like algorithms for biomarker models, essentially deriving these equations or algorithms. Okay. So this is another personal example of mine where we had looked at two different subsets of asthmatic individuals. And this graph is just showing you how they respond to inhaled allergen. And you can see there, this measurement here, forced expiratory volume in the 1 second, is just a measure of their lung function, how much they can breathe in the first second of their exhale. And you can see it drops right away once they inhale the allergen they're allergic to. And it recovers naturally. However, some people have different responses, right? So some people recover and some other people have another response later on. And you can see after you run some biomarker pipelines and identify these molecules and sort of attribute them to pathways, you can see that many of them may actually belong to certain pathways that are very prominent in driving these specific responses, right? So you can see, instead of capturing a single molecule that's sort of your biomarker, you have essentially a whole team of biomarkers, right, that is actually giving a much stronger signal in discriminating your groups of interest. Although it's very useful to have a panel of biomarkers, I just wanted to have this like pros and cons table for why single biomarkers may be better than a panel or the other way around. And so single biomarkers have a lot of advantages in that they're a lot easier to develop, right? You're essentially developing a single assay, and the chemistry is a lot easier to implement in the clinic. There's already like a lot of examples in real world settings where you're just looking for a single marker, and it's a lot easier to interpret for clinicians. The cons are that they're obviously not perfect, right? So a lot of biomarkers that are out there are actually very generic, and they can be modified by a lot of factors, whether it's by disease or environmental and so on. Panels of biomarkers are much better at capturing the the heterogeneity of the disease. And there are some examples of multi-panel biomarkers. There's one for breast cancer, and there's a FDA-validated nanostring assay called Prosigna that essentially is a 50 gene panel that can stratify different subtypes of breast cancer. And the real world data we'll analyze has some of those subtypes there. The cons are, again, it's not perfect, right? You're not capturing the entirety of whatever is going on, and that could just be the biology is like pretty complicated, right? So using, for example, just proteins or RNAs may not capture the entirety of the picture, and that's where like multi-omics may come in. But then the problem there is that developing a analytical like blood test that contains many of these markers becomes a lot more challenging, right? So you sort of have to weigh the pros and cons in terms of what's your shortest route to the clinic and what is like your low-hanging fruit and what is the patient need.
I have a question.
Okay.
So when it comes to using multiple biomarkers, I guess statistically you'd need a larger sample size if you're going to start including multiple variables to separate two groups. Is that true?
Well, that was true in the past with a lot of the classical methods, because they couldn't handle more variables and observations, but a lot of the newer approaches can handle like high dimensional data through something called like regularization. And what happens there is that it shrinks a lot of these weights on these variables, so they're not like inflated. So the new I guess age is really data centric. And so what that means is you want to have exquisitely like phenotyped individuals. And so it just makes it easier for your algorithms to separate them, right? And so that's really the main I guess distinction I would make. Although during some of my case studies, I'll sort of go over like the real like case that most people are in, where they have like a small data set, but they have so many variables they've measured. It's like, how do you estimate the test error, right? It becomes challenging. So I will show an example of that.
Okay, thanks.
All right, so some more terminology, because again, humans have heterogeneity, we have diversity, so people use slightly different vocabulary. So I use the term biomarker panel to refer to two or more like molecules that sort of go into a model or an algorithm or an equation, right? And that's sort of what your biomarker panel looks like. May I ask if Amrit could share the paper? Oh yeah, sure, I can do that. And so there's a lot of terms for it. So biomarker signatures, multi-marker panels, biomarker models, algorithms, like anything you can think of, where you have like more than one molecule or like more than one variable, right? So sometimes biomarker algorithms could have like sex or age or an image, right? So all of these like different types of variables can be combined into one algorithm. Then we have different notations for the cohorts that we use, right? So in a lot of the biomedical literature, we have something called a discovery cohort, and that's essentially the initial data set that's used to develop biomarker panels. And then we have a validation cohort or cohorts where you essentially test your panel on these independent data sets. In the statistical or like computer science literature, we have these cohorts referred to as sets. And oftentimes we have a training set and a test set. And the training set can be further divided into multiple sets as well. So like training set, validation set, and so on. And the convention varies a bit as well. Technical replication, it's the defined as confirming your findings in the same set of samples. But you're sort of using a different technology. So if you identified like a biomarker using RNA-seq in a set of cohorts, then if you sort of look for that protein again in the same people, but using like say another technology like RT-qPCR or nanostring, then and you validate your findings, that would be considered a technical replication. And so you might say, why would you want to do that? And oftentimes that transition step between an unbiased platform like RNA sequencing to a more like clinically relevant platform can have significant impact on your biomarker performance, right? So there's a lot of unbiased technologies that can be very less sensitive and specific. Whereas once you start going into the clinical chemistry, you have to be very specific in what you're designing your test for, right? So the technical replication is actually very important. Thank you, Kang. And then validation is just confirming your findings, but instead of the same cohort, we have some samples from the original. So this is using an independent cohort. So I think this should be modified. Let's see. Can't spell today. So one other note that I want to make at the very bottom is that based on my experience in terms of how to actually test algorithms in practice, the deep learning literature that if you ever do courses and stuff, there's often this concept of a train, validation, and test sets. And so oftentimes the train and the validation are what are used to sort of optimize the model. And so you can think of the validation set as consisting of observations that really mimic the test set and is what is going to eventually be used in practice, right? So for an example, if you're looking at some sort of like an image classifier to detect like cancer nodules on CT scans, for example, right? The training data set you can be quite lenient with. So in terms of you can like take a bunch of pictures from people, you can rotate them, zoom in, zoom out, and all that kind of stuff. And really try to make it hard for the classifier. But you're actually tuning the classifier based on a cohort that's actually representative of what you want to ultimately use the cohort on. So suppose if you want to use your algorithm on a targeted population, then these sets are going to be representative of that. And this model that's being trained will sort of fine tune itself to be more specific to that population of interest. In the biomarker literature, there's often different terminology. So instead of like train, validation, test, which is like under the hood still being used, we have like a discovery and validation cohort. And the discovery cohort is what is really divided into a train and validation set. However, there's this concept of something called like cross-validation, where essentially it's a process of dividing your discovery cohort into multiple train and multiple validation sets. And we're going to see examples of that as well. All right, so how do you identify biomarkers, right? So there's a lot of approaches that people have used to identify biomarkers. And so I've listed a couple here. So you can do like differential expression, right? Use a cutoff, find like a bunch of molecules, and that's your panel of biomarkers, right? You could use machine learning methods. You could use a priori list of biomarkers. You can use a combination of them, right? So the easy part is actually coming up with the list of biomarkers, right? If you have a data set and you can apply some of these methods, then obviously you've found some biomarkers. But the hard part is actually determining the test error of those biomarkers, right? If you don't have additional data, then what are the techniques you can use to determine whether something is a biomarker, right? So suppose you identify a list of molecules, but when you estimate the test error, it's like 50% is like random chance. Those are not really biomarkers at that point, right? And so this is what this session is really about. It's the techniques used to actually determine the test error in the absence of additional data, right? So you have one cohort only, and you have like one way of determining these biomarkers, and how can you find out what the error will be in samples that you have not yet recruited or seen. And so we're going to look at two case studies to sort of apply some of these tooling to. And the first case study is a real world example on breast cancer data. And in this case, we're lucky we have two cohorts. We have a discovery and a validation cohort. And this cohort is 150 samples, patients, across 3 groups. And there's a training set, and then there's also a test set. So the training is referred to as a discovery, and the test is referred to as a validation. And then I have a case study, which is on one cohort, and it's on simulated data. And this is where I'm going to show you how to estimate test performance on in the absence of additional independent cohorts. All right, so let's look at case study 1. So here I've just, any questions so far? Okay. So here I've essentially come up with some hypothetical approaches that one might use to come up with some biomarkers, okay? So here I've done a like a pipeline, you could think of like a series of steps where first a differential expression analysis was performed. And so here I've basically just taken the breast cancer training data for the mRNAs. And since I have 3 groups, I'm doing an ANOVA for each of the individual mRNAs. And then I'm recording the p-value. I'm adjusting the p-value based on this is Benjamin Hochberg. And I'm keeping the first 50 molecules that have the lowest FDRs. And so that is my my biomarker panel, right? And essentially to come up with an equation, I need to sort of put it in like a logistic regression format. And I'm using this analogous model called the partial least squares discriminant analysis. And you can sort of think of this as a logistic regression equation. And but the neat thing about this is that similar to a PCA, which maximizes variance of your data set, this approach maximizes covariance of your independent data matrix X and your response variable Y, right? And that's what any model, like a logistic regression model, is doing. However, it actually computes components as well. And so you're able to get these similar looking clustering plots, but the clustering is actually supervised by this outcome variable Y. And here for the 50 genes that were identified, I'm plotting the clustering patterns of those 150 individuals that are part of the training data set. And you can see already by the expression values, we have like the individual groups, the 3 groups being clustered separately. And the groups are basal, HER2, and luminal A. Any questions so far? Okay. All right, so our second method that I'm still applying to the breast cancer data set is to forego the differential expression step and just apply a model that actually does something called variable selection, right? So these new, I wouldn't say news, the lasso was invented in 1996 by Tip Shirani from Stanford, which essentially applied a penalty to the loss function, which allows you to select variables by shrinking most of the variable weights to zeros, right? So in that case, you actually get models where there's only few variables that have non-zero weights. And this is, again, another model that's analogous to a variable selection with like logistic regression. So this is called the sparse partial least squares discriminant analysis. And here, just to keep things similar from the previous step, I'm using a model that has 50 molecules in total. And so you can see here, this is a totally separate approach, right, from the first approach. And I've gotten a similar sort of clustering, similar sort of separation. And so you can think of this as like two labs doing, they're using different approaches, and they have like two different biomarker models, okay? And you can actually do an overlap between them. So this is kind of hard to see, but essentially you can think of these two circles as the two panels, and there's like a 43% chance overlap. 43% feature overlap, and there's obviously some uniqueness in the approaches that's used, right? So that's all good and dandy, and a third lab can come and do another analysis and find another panel, right? So you can see that there's a lot of panels that can be identified using various approaches, right? And so the key is, what is the performance of these panels, right? So right now we're only just like identifying these panels that are arising, but we have not spoken about the performance of the panels, nor have I touched on what is the correct number of variables to select, right? So that's something I want to touch on later on. All right, so let's talk about really evaluating the performance of these biomarker models, okay? And so there's a lot of performance metrics out there, and you can see this Wikipedia link. And this has a lot of measurements here. And so these are a lot of metrics that can be used for biomarker or like any machine learning models pretty much, right? There's a lot of them. And so here, for the sake of this talk, I've only listed a few common ones. So the area under the receiver operating curve is for a binary classification. It's a very useful measure that depicts a trade-off between true and false positive rate. Mean squared error is useful in the regression context where your outcome variable is continuous. And so essentially you do a subtraction between your observed actual, and you square the differences and sum them. Accuracy is essentially the proportion of correctly classified observations. And so this is also a useful measure. F1 score is, again, combining multiple performance metrics to sort of get a trade-off between these things. So there's a lot of different things we can use to evaluate performances as well.
Hey, Emre, so thinking a lot of machine learning, isn't there a lot of debate about how the imbalanced data sets can give you beautiful AUROC values? So don't people mostly now use the area under the precision recall curve?
So the problem with the imbalanced class sizes, there's a bunch of ways to handle that problem. So I talked about accuracy, right? Which, if you have like imbalanced class sizes where, say, like 90% of your a cohort was like one class and 10% was another, and you did really well on that cohort with 90%, then your accuracy is going to be really high, even if you missed all of the 10% people, right? And so
Yeah.
you can do things like balanced accuracy, where you actually compute the accuracy individually in the individual classes and then take an average. You can also do something called a weighted what is it called? Like weighted residuals in your model. So what happens is you can so the models, if you look at the loss function of these models, they're essentially doing some sort of averaging on how far away the predictions are, right? But if you have more observations of a given group, then you can actually downweight the more frequent class type and sort of upweight the minority class type. And so you can actually incorporate these type of observational weights in your models as well. So and you can also do like sampling as well, right? To like balance out the imbalanced class sizes. So there's a lot of approaches that you can use to target that issue. And so for the purpose of this talk, if you remember from the cohort description when I talked about the breast cancer data was here, you can see that there's oh, it doesn't show you the individual allocations, but I can actually just read in the data set here. So if I show you how many subjects are in each group, right? So this is like luminal A has double the amount of HER2, right? So if I did really well on luminal A and I totally missed these, I would get a reasonable accuracy, right? So some of the code that I have done is around like balanced accuracy, where I compute error rates separately and then take an average. So there's a bunch of ways you can handle that problem.
Okay.
Amrit.
Yes.
I have a question about the accuracy. Is error rate pretty much synonymous with accuracy?
Yep. So if I go back to the table here, so you said accuracy and error rate, right? Yeah, so they're essentially mirror images, right? So the more you get right, the less you get wrong, right? Or so you can do 1 minus the accuracy and that'll give you your error rate.
And the other question was about the group sizes. If you have the same group sizes, then safe to say that the accuracy would be unbiased.
Yeah, you could say that. You could say it's less. I wouldn't say unbiased, because bias and variance are very different. There's actually like some theory around those concepts and how the models perform, but I would say they would be less I guess affected by class imbalance.
Thank you.
And I'll speak to a bit about bias and variance in a bit. It just relates to how well your model is, how far or close it is from the truth, and how variable are the predictions. So the concept of bias and variance is very important in machine learning. So let's go on to the next. Okay, so now we're going to start determining the performance, right? And we're actually very lucky in this first case study is that we actually have test cohorts that we have not seen. And so for the two different methods that I use to determine biomarker models, the first method, when I apply it again to the training data, right? So it was built using the training data, and I reapply it back to the training data, and I get an accuracy and a balanced accuracy of 96%. And you can see the calculations here, right? So the model is called diff model based on differential expression. And I'm putting in the training data set back in the model that was trained on. So you can already imagine the accuracy should be really good. And then I can compute something called a confusion matrix. And it looks something like this. And you can see along the diagonals are where we have gotten our predictions correct. And then based on the confusion matrix, we can compute the accuracy, which is essentially everyone we got right over all of the individuals, like tallied up, right? So there's very few individuals that this model got wrong. And this is like 6 individuals it got wrong. But this is expected because it was built using the same data, right? Similarly here, So you can see the results are exactly the same, right? Regardless of the two approaches, even though there was differences in the model features. But ultimately, you want to have an independent cohort. So in this case, we did have a test cohort. And so when you apply it to this independent cohort, you can see that the performance usually drops considerably. So in the diff model here, the performance, and if I show you the code for it, the model, let's see, left. Okay, so the diff model is here, and this is the test data set that I'm predicting the labels for, and this is the confusion matrix there. So you can see the accuracy dropped, but so this is a purely like formulated data set. So I had essentially like created this data set, but it had over 1,000 individuals, so it was obviously cherry picked somehow. I don't know who did that. And created these train and test data sets. So they're obviously cherry picked to be like really good performing. And you can see that even the method 2 model, the performance is really well, right? So in this scenario, it doesn't really matter whether you're using these methods. Both models are like valid because you've tested them in independent cohorts. So in your publications, you would report like this accuracy in your independent cohorts. Any questions so far? Okay, perfect. All right, so I think most of people in this room or in general don't actually have that extra cohort, right? It'd be great to have thousands and thousands of cohorts and patients, but that's not the reality, right? So what is the strategy to estimate the performance in that scenario? So the answer is to really split the data and pretend you do have test cohorts. That's basically the best strategy that we have, all right? And so there's different schemes that you can use to estimate the performance of your model. So the most common one that you'll probably run across is something called k-fold cross-validation. And essentially it's a strategy to split your data into k-folds of equal sizes. They can also be stratified based on groups. And k essentially is a number, right? So if your data set has like 10 observations and your k is 5, then each fold or each split of the data will have 2 observations, right? And so you can see that you're sort of recreating this train and test scenario just by splitting your data into these multiple folds. And I'm going to go over exactly how this process works in the next slides. And so there's this k-fold cross-validation where you sort of go through your different folds. So if you've created k-folds, for every fold, you have to train your model on k-1 folds and then test on the remaining fold. And you can basically repeat it until you've exhausted all of the k-folds. And then you get a performance metric out. But this can be repeated multiple times just for stability of your metrics. And this is often useful in like small data set scenarios. And this is another much more complicated scenario here where you actually need to determine basically the optimal model to use in practice, right? So oftentimes there are a lot of hyperparameters that need to be tuned. For example, in the SPLS-DA model that I had arbitrarily picked 50 molecules, right? 50 features to select. And so you can actually use tuning. So you basically use a grid of values to select your best model, but that process needs to be cross-validated as well, right? So within each of your cross-validation folds, you need to actually do another cross-validation. And we're going to see an example of that as well. And here's that little note about bias and trade-off. So this is very useful when you're actually choosing the value K, right? So if K equals N, that means that every fold only has one observation, right? So its bias is going to be very, it's going to be on the lower side because it's using basically all of the data to develop the model, right? But its variance is going to be a lot higher just because for every given fold, only one value is kept out. And so it can be very different from the rest of the data set. And so the recommended values for K are 5 or 10 based on the literature. All right, so now the like I went through the cross-validation very quickly, those definitions, but it's actually very important to go through it with a little bit more detail and higher resolution so you actually understand exactly what's happening within this process and what is actually happening within each of the cross-validation folds at every iteration. Okay, so here I'm giving an example of k-fold cross-validation. And in this case, I've sort of turned the matrix on its head. Oftentimes the observations are on the rows and the variables are on the columns, but I've sort of transposed the matrix just so it's easier to view. And I've elongated the width here just to show that there's n observations, and it's just easier to visualize once I start breaking it into these different folds. Is everyone following me so far? We have an X matrix, and then for each of the n individuals, we have a response values, and these can be continuous or categorical. Give me a thumbs up if you're following so far. Actually, I can't see, so okay, I get some thumbs up here. Cool. 5 thumbs up. Oh, cool. It tallies it up for you. Nice. Okay, so that's the first step, right? We have our data set, and now we're going to go to step 2, which is to divide your data up into folds. So for this scenario, I'm using a 4-fold cross-validation, right? So I've essentially stratified my data set into 4 pieces. And so each piece has n over 4 observations, okay? And I've labeled them as fold 1 to fold 4. And now the fun part begins, right? So now we actually hold out one part of the fold. So sorry, one fold that we hold it out, we call it test. And the remaining 3 folds, we call it train. And so this is very useful now because we sort of recreated that train test scenario we had talked about, where we sort of develop our model on the train and test on the test set. And then so here, we're now using the training data set to develop a model. And so this is where things can become a little bit more nuanced. So I want everyone to pay attention to this detail very carefully. So oftentimes when people think about creating a model here, they're just thinking about like the final equation, right? So applying like a machine learning algorithm to train. But this model circle that you see here really refers to the model development process, right? So there's a lot of things that can go that can be involved in here, right? So this, when we talked about differential expression, selecting candidates a priori, machine learning algorithms, all of those can be combined in this step here, right? So all of them needs to be repeated in this training fold here, and you come up with a final model. Once you've come up with that final model, regardless of how you do it, you need to then test it on this held out test set. And then you get a set of predictions out, right? So these could be like predicted continuous values, or they could be probability predictions and so on. And you essentially then repeat this procedure. So then you sort of use the next fold as a test set and you rebuild that model. And then you apply the model to that held out test set, and then you add these new predictions that you just created. And you keep repeating this process, And I couldn't find figures on this process online, so I created these figures for you. So if you need to see these figures for yourself, you can actually find them in the image folder under CV, and the images are there, and there's a PowerPoint slide that you can use as well. And so ultimately what happens is at the very end, you have applied this model building process to all of those training sets, and you've applied it to the test sets. And now you have essentially a vector of probabilities or numeric values, whether if your response was continuous. And now you have like two sets of vectors, right? One is your like truth and one is your predictions. And now you can actually compute your metrics like AUCs, mean squared error. All right. Any questions so far at that stage? Is this clear, the cross-validation step? Okay, perfect. So let's now go to the simulation scenario, because this is where things are going to get interesting, and hopefully I'm able to convey why this is so important. Okay, so the case study 2 is the case where we have a single data set. Okay, so here I'm generating a data matrix. This has 100 subjects, and it has 1,000 variables. Okay, and the 100 subjects are divided up into 2 groups. Again, they're equal, so there's no that imbalance class size problem. And essentially I've created the data by randomly generating like normal values with a mean of 0 and standard deviation of 1, right? So you can see here that this gene expression data set that I just magically created is just random noise, right? And I'm just randomly allocating them into these 2 groups here. So each group has like 50 people, okay? And I have gone ahead and done a t-test for each molecule or each feature in my gene expression data set. And I've come up with a list of p-values. And then I've ordered the p-values, and I picked 10 molecules with the lowest p-values, and I have a list of biomarkers. Yeah, this is a good question here. The model it will be different every cross-validation fold. Yes. Okay, so now we have gotten our list of biomarkers. There's 10 biomarkers. The p-values look great, right? And we can actually plot the p-values here. And so if anyone's not seen a graph of p-values, this is the p-value distribution when the p-value actually follows the null distribution where nothing is significant, okay? But in the real world scenario and the real world like published literature, unfortunately this happens where people will do an analysis like this, and we have these biomarkers of interest, okay? And they have identified these biomarkers, okay? And now, as I mentioned, you have a list of biomarkers, but you must report the error rates for these biomarkers, okay? So let's first see the wrong way to do cross-validation. Okay, so, and unfortunately, this is actually very common in the literature, right? So if you see this, I would recommend that you flag it right away. And so the wrong way to do this is to actually take those list of variables that you identified. And so in this case, we have our gene expression data set, but people may say, hey, we have sort of reduced the noise in our data set by selecting features that are associated with the response. So now we have a revised list of molecules, okay? And then we sort of build our model using those list of molecules. And then we compute our cross-validation using M-fold cross-validation. And there's 5 folds in this case. And what is the area under the curve? And the area under the curve is 0.87, right? So they literally created a Biomarker panel. This is like really good AUC, right? And now you can publish your findings. Okay, but we know what the true error rate. The true error rate here was 50%. Right, so this data set has no signal, yet many people report that it has very good signal. Okay, so this is a big X over here. And unfortunately, this happens a lot. The right way to do cross-validation is to not do this cherry picking of the data. Okay, so you actually need to use your entire data set when you're creating the your model, right? So this model here has a has 10 molecules, right? Same thing as this wrong model. This has 10 molecules. This also has 10 molecules. But the performance of this model is determined by cross-validating the entire data set. So you can see that the question that was asked is that model different between the cross-validation fold? It's like, yes, that model is actually very different because that's the whole point of validating this model building process, right? So you can see here that the correct way of doing cross-validation, we get an AUC of 0.5. Any questions here?
You said you see this in the in the published literature?
Yes. And I have sorry, my video's off. I actually see this. I've even seen this in like seminars where that's the advice someone is given, right? It's like you wanna like filter your data set first before you do this, which is actually I'm gonna actually show you how you can filter your data set, but do it the right way, right? So here I'm showing you not to subset your data set, but some people may be adamant in terms of really wanting to include differential expression, right? Because sometimes they want the fold change, for example, to be an important consideration in the biomarkers that are selected, okay? And so the way that you want to include that in your approach, which is my next slide, is within each of the cross-validation folds, you start with all your data, perform differential expression, then use the top features to build a model, and then apply to the test out set. Okay, so basically what's happening here is that the approach that was used to develop your final model needs to be repeated within the cross-validation folds. Okay, so if you remember the way that we actually found our the model of interest was up here, right? We had done a differential expression here, and then we had found a model here, right? But the way that we estimated the cross-validation was wrong. The proper way to estimate the cross-validation for that approach is the following. So this is slightly more involved here, so I have outlined it here. So the first thing you can do is create your cross-validation folds. So I've taken the list of individuals, right? And I put them into different folds, and there's 5 of them. And then for each cross-validation fold, I create a training and a test set. And then I perform differential expression, and I perform the model building process as well as predicting on the test data. And then I get a list of predictions out because I've actually repeated that model building process within the cross-validation fold. And I get a list of predictions out. And that is what I'm using to compute the AUC. And you can see now we've actually gotten the correct AUC, which is 0.5. So this is can be very nuanced, right? Because people may not understand why this is important. So if you have questions at this stage, please ask.
Has anybody written, I guess, common pitfalls in evaluating models? I mean, I guess this would be kind of something that would be key among them, but is there an aggregation of all these different issues in a like a review or something?
Yes. So I might not have it here. Let's see. So there's a course by in Stanford. It's called Statistical Learning. And the people that wrote the book on element of statistical learnings have this outlined. Literally, I learned it from them. That's why I'm talking about it here today. And this was back in 2014 when I first learned it, right? So this is a talk by the Professor Trevor Hasty. And so he does go into the right and wrong way of doing cross-validation because they've also seen it in their work as well, right? So here, this is the wrong way is where you select your predictors and you do cross-validation on your predictors. But the right way is to actually do cross-validation on all of them. And so the reason why I've created my slides slightly different is because a lot of people will miss the nuance of the model building stage, right? So this stage here, people will miss this nuance. So they think that the filtering and things like that is actually not related to the model building process, but is actually linked directly to it because you've literally peeked at your data before you build your model.
Cool.
Yeah, I see a question here. Do you then report all different models that you have the top features in your publication? No, you don't, because what you're actually reporting is your that one model. Actually, you're reporting this model, and there is like some controversy here, but you can I can show you if you want to report all of them. There's a way to do that as well. So let me first show you the first approach. This one here, right? So where we had these models, right? So this was our final model where we had these 10 biomarkers, right? So the performance of these 10 molecules comes from the cross-validation performance, right? So we don't need to worry about what features were selected within those things because we're actually just validating the model building process here. We just care about the error rate, right? So the error rate we get out of that model building process refers to these 10 listed molecules. Okay. However, now, if you did, and this might be like a little bit more complicated. So if you took this model here when you did the cross-validation, right? So I think Mingold is referring to this, right? So when you get like different lists of biomarkers here. So if you went through this approach and you got like different lists of biomarkers, you could look at like stability of molecules, how stable are specific biomarkers over others, and then you can sort of report maybe like this biomarker always shows up in all of these fools, where these other ones are more variable, right? But this performance that you see here refers to this list of biomarkers. Is that clear? Okay, perfect. All right, now I want to take it slightly more complicated. And I'm glad I went through that really fast. So I don't have a lot of slides left, but this is probably the most complicated of the topics here. So now we're talking about something called nested cross-validation. Okay, so what's happening here is that within each of those training folds that we sort of talked about earlier, there could be an entire another cross-validation that's occurring. And the reason for that could be something called hyperparameter tuning, right? So I talked about SPLSDA, which is a method to discriminate two or more groups. You can think of this as like multinomial like regression classification. And so this is again a supervised analysis. And this is coming from the Mixomics package that I contribute to. And in this case here, we have this argument called keepx. Okay, and so a lot of algorithms that include something called regularization may include these penalties that allow you to select variables. And that becomes important in the interpretability, right? Because if you don't want a model that has like 200 molecules or like 1,000, it's harder to interpret what that means. Whereas I guess the key innovation of this package is that instead of using penalties, you can actually specify a grid of values to retain in your model, and then you can actually select the best model that has the least error rate or the highest accuracy. Okay, but again, it's slightly complicated because you sort of need to repeat that process within each of these training folds. Okay. All right, so going back to Mixomics now, there's a function in Mixomics called tune, which allows you to feed a grid of values, right? So here, if you remember initially, when I was applying these models, I had arbitrarily picked like 50 genes to build my model with. Now I can actually specify a grid and test all of them, right? And now here I'm using the breast cancer data set again, discriminating between three subtypes, five-fold cross-validation. And you can see that for every given keepx or number of features selected, I have this error rate that's being recorded, right? And so based on this curve, I can select the optimal model. So maybe like this model here has 50 genes that gives me the lowest error rate. Okay, and so the final model now is developing the SPLSDA model, but retaining the hyperparameter that was associated with the optimal model based on this process that I had used. All right. But the question now is, now I've determined this model, right? What is the performance of this model? Can anyone care to guess how we could estimate it?
Before moving to guessing it, I do have a question about the tuning step.
Okay.
So it looks like you pre-selected the number of components to be 1. So it sounds like you're basing this tuning on only one component. How did you come to that determination?
That's just for the sake of simplicity, but that's another thing you could tune as well, right? You could use a grid of components, and you can get multiple curves for multiple components, and you can sort of study that as well. The more components you add will often lead to lower error rates, and just for the sake of simplicity, I just stuck with 1.
Sounds good. Thanks.
Any other questions? Yeah, just looking at the examples here, I feel they also used 1 here. Well, this one is a tune method, and this one has 2, right? So you can see if I just run this, since we have time, what it looks like. So if I plot this output, and repeat as 1. Okay, sorry. So seems like there's a problem whenever they need to compute the standard deviations. Maybe I'm not specifying the argument correctly. Let's see. Oh, that's correct. I'm not actually sure why that is. I'm gonna try the breast cancer data set again. Okay, so this is, you can see, speaks to your point a little bit, or I could do it here as well, right, for the data set we're working here. So if I do 3 and just like run this, keep X must be non-negative. Oh, this one here. Okay, so let me actually just highlight this out. Okay, so again, this and repeat thing is causing issues. Okay, so that ran, and if I plot the result, so you can see here I've essentially added more components, but it was complaining because it couldn't plot the results because it couldn't complete the standard deviation. So I just added some repetitions. We'll actually repeat this process like 5 times. And now we have some standard curves. So you can see that when you retain one component, then this is what the error rate looks like as you select features. And then if you have more and more components, the error rate drops significantly. And essentially just adding more and more independent variables, right? These components are being added. And so the performance is dropping, right? But the problem there, I would say, is that now you're retaining like too many variables, right? So now you retained, this is per component, so you retain 300 variables. So ideally, you want to favor towards less variables for biomarker discovery. So maybe even something like this model here. Sorry, there was a hand up. How do you know when you're overfitting? Okay. I would say overfitting, you would know when you're overfitting is when your test error is like really bad. So if it's like 50%, you know that your training model doesn't generalize to the test error. And the examples that I've shown you here, they've been quite, I don't think I've done, they've been quite, I guess, like cherry picked in terms of the performances are quite good, right? And so the only example where we've overfit the data is the first one, where the performance was really really good. Sorry. Oh, this one's going. Yeah, actually, I would just keep it at that. You've overfit your data if your performance in your test set or in your cross validation is really bad. And so this cross validation, when done correctly, can actually in most cases it actually will show that your model contains no signal, right? So oftentimes when I've seen cross validation done, it's usually done the wrong way, like this, where they've cherry picked their features. But if you've followed the correct methodology to compute your error rates, then you will get an appropriate estimation of the test error. And that's basically your best guarantee in terms of your model not like overfitting, right? So I guess there's like two strategies that are being used to combat overfitting. The first one is cross-validation. And the second one is the concept of regularization, right? So the penalization that we're applying to the residual weights makes our weights much more stable. And this actually helps them from overfitting the data. Hopefully that answers the question. All right, so going back to the, are there any other questions before I go back to that last part of the talk?
Hi, Emery, and can you hear me?
Yes.
And here is that how to do in these slides. In this slides, just you showed how to do the cross validation, right? And here I'm still a little bit of puzzling, even if you explained that before. So you mentioned that in the right way of doing the cross validations, and each time we split, for example, the 5-fold cross validation, and we divided our samples into 5 parts. 4 parts are utilized to make the right model, and 1 is test. Then the next time, and another 4 part of data are utilized to train the model. In this case, the first time, the second models may be different, right? And so after the running of the 5-fold cross validation, we have maybe 5 different models, and then as somebody asked before, and once we are trying to report our model, we report this 5 models, or we just report 1. I'm still a little bit puzzled about that part.
Yeah, that's a good question. So I think the reason why it becomes confusing is because we're very focused on the molecules or the features that are selected, right? Because that's sort of our end goal, is like, what are those biomarkers, right? And so as soon as the those things start changing, we get a little scared. But instead of thinking about those feature selection, we should think about the model process, right? So even though the model is different within each of the folds of the cross-validation, the final model is developed on the entire data set. Okay, so if you look at this with that wrong and the right way that I've done it here. Sorry, not this one. This one. So this one here, I had taken the entire data set and I had chosen 10 features to develop my model with, right? So if I run this model, so here, if I show you what this model looks like, so let's see, we do select var. So this is what that model looks like, right? And this model was actually applied to the entire data set, okay? But now we actually need to find the performance of this model. But this model is related to this process of identifying the model, which is applying this machine learning algorithm, right? So when we do cross-validation, we just apply the same process every time. We don't care about which molecules are being selected and so on. We're just more interested in applying that same process every time within the folds and get a prediction out. And the reason why that's important is because in the real world setting, you're gonna have like different models that are being developed, right? If you had like different cohorts, you're not gonna have the same model.
Okay, but after we use the right way to do the cross validations, and I mean here, because we are using some random produced data, of course the UC is not good. But under the conditions that in some real research project, and after this right way to do the cross validation, we get a very good AUC, for example, 0.9, and then we are getting ready to write a paper. And here we didn't report the models, right? We just reported the features that we get, and these panels are the biomarkers. Am I right?
Yeah, so this would be your panel.
Okay, yeah. So here we are not focusing on the models. We generally focus on the panels that we can discriminate the different classes within this data set.
You have both. You have the model and you have the error rate.
Okay.
Yeah.
Okay, I see. I get it. Thank you.
Yeah, so this is a slightly tricky bit, right? So you might have to not worry about like the models and the features that's happening within the cross validation, because obviously they're going to be different.
Okay, I see. I get it.
It's going to be
Yes,
confusing because then you're going to be worried about like different molecules that start appearing in different cross validation folds. Yeah.
Okay, yeah. So before I'm really confusing, but after your explanation, I got a point. Thank you.
You're welcome. It is right to say we're valuing the model or present on the model. I would say yes to this. And the model we generated during, you don't, I'm not sure if you're not generating one model in the cross validation, you're generating multiple, right? So this is where my next case study will actually make it even more clear. Let me scroll down. Okay, so I'm going to go back to this example, right, that I was showing you. This is where we had, I'm going to save this, so then it'll run and it'll update this part. Yeah, there you go. So if you remember the first approach we had used, we did like differential expression plus model building, right? Now we've gone a totally separate route, and we've actually took our entire data set, we applied this entire procedure, and we've come up with a final model that's based on the number of features, which is related to the model with the lowest error rate. Okay, so now we have this optimal model that has 50 features. Okay, and now we still don't know what the error rate is for this 50 genes, right? It is not this. It is not 10%. Can someone tell me why? And the reason, okay, so the reason why it's not 10% in the error rate we're going to see in like new people is because we've literally tested all these different models on the same data, right? So I can literally start randomly picking molecules, and I can give you a model with like 0 error rate. Okay, and this whole process is a very analogous to the process we did with differential expression, right? So what actually needs to happen when we're trying to, again, determine what is the performance of this model in like data we have not seen yet is to repeat that same cross-validation, but within each of the training test folds, we need to repeat this process. Okay, so to get with the panel of the performance of this 50 gene panel, we need to go on to the next slide here, which is estimate test error using only the training data. Okay, so again, that same process that I had talked about earlier. Sorry about that. I can't scroll back. Oops. Sorry, I'm on a Mac, so it's I don't know why. Okay, sorry, I'm just gonna go in my browser, make it easy. So if you remember from the differential expressions that we had within each of the folds, we had done differential expression plus model building. Now we're actually doing that same line of thought, but within each of those steps, we're actually tuning the best model and finding it out. Why do I keep doing that? Oh, sorry, frustrating here. Within each of the cross-validation folds, we're actually finding the best model here and then applying it to the test data set and then getting a list of predictions out. Okay. And then using that strategy, we have an accuracy of about 87%, or a balanced accuracy of 0.86. So this is, if you remember, the accuracy is just 1 minus the error rate, right? So the previous slide showed an error rate of 10%. I was showing that's not the error rate of this because we've tested 1 billion models. In the true, like, in additional samples we have not seen, the estimated error rate will be 13% or 87% accuracy, because we have done this in an unbiased formulation here. So that performance for this 50 gene panel is 87%. Okay. Any questions at this point? Because this is a very where I think I might lose people, because I'm sort of talking about this model process validation. Can you give me a thumbs up if this is clear? How we're sort of validate the model process here and not trying to like cherry pick our data, right? So whatever you use to create your model, right, needs to be within that cross-validation when you're validating it. So in this case, we used hyperparameter tuning, found the model with the least error rate, and that was our final model. So we have to repeat it in the cross-validation. All right, so that was the error on the training data set. So now let's identify what is the error on the test data set, okay? Since we have that available, we're very lucky. So let's apply it. So we took our final model. This was the model that we had done the 50, we identified those 50 genes, right? And we applied it to our test data set. And now we have an accuracy of 90% and a balanced accuracy of 88%, right? So this is the accuracy in the independent data set. And this is the accuracy of our estimation of what it would be in the independent data set, the correct way. Any questions so far? Are you surprised the accuracy is higher in the test data set than the training? No, because this is actually a very like cherry-picked data set. I don't even remember whether the what it was in the paper. Actually, let me check. I'm not surprised. What you want to actually see is it needs to drop in the test data set, because obviously like the training data set is not going to be the complete picture, right? So this is the actual paper where I had created that data set. It's this one here, breast cancer. Like there's 989 people. Oh, this might not actually be useful for this purpose, because this is multi-omics. I'm using all of the variables. Never mind. But I should have listed the error rate somewhere. And we have time, that's why I'm slowed down a little bit. So you can see here the classification error rate is between 11 to 28%, or 19 to 21%. So it's a similar range that we're finding using one data set. Maybe this is the most predictive of them all, right? So if I use like microRNAs or CPGs, then maybe I had gotten a lower performance or something. Okay. So in real world settings, expect that the performance and the correct way of cross-validating to drop significantly. So then the last slide that I have here for today, after giving this like controversial presentation, is should you biomarker? And so I have this image, it's not rendering, so let me just knit it again. And so the reason why I like doing cross-validation the right way is because it's actually much better way of assessing how predictive your biomarkers actually are, right? Because you can get very predictive things using multiple testing correction, using like robust statistics and so on. But this cross-validation process, when repeated properly, will actually, oftentimes in my case, it actually has helped me to nullify a lot of findings. So I highly recommend you follow this process of estimating your performance and not be too bummed out about not having the best performance for your molecules. It just tells you that there's not enough signal to discriminate your two classes. And that's pretty much what I had for the slides. And I think I have some resources if you want to check it out on cross-validation. And thank you for attending. Appreciate it on Friday afternoon. I'll take questions.
Yes, thanks, Amrit. I'll echo Mangold's remark that thanks for going through this. It's really helpful. And I think for me to raise more questions than, well, I did raise a lot of questions, but one particular one for my personal application was about repeated measures. How does everything you've explained, how does that translate to a repeated measures design where you have an intervention and you have the same population and just seeing them after different interventions? How does that change any of the things that you've done?
That's a good question. And in the Diablo paper that I had was talking about, I had a similar like repeated measures scenario. So this data set that I had is an asthma data set. It has like a pre and post scenario where the individuals are the same in these two different time points. And there's this this like type of analysis called multi-level analysis that allows you to it's analogous to like taking differences between like subjects when you're sort of computing like effect sizes, but it has this reflection along the zero axes where you have like negative and positive numbers. And essentially the way to handle a pair design as such is to keep your like within the cross-validation step, you need to not look at your test slides, but use your training data to normalize your test data set, right? So you can normalize using the averages and means and things like that. And that will basically scale your observations to those measurements. And that's one way of how especially mixed omics can answer some of those like paired measurement questions. So you can see here, I have, this was done a while ago, so I don't actually remember the difference between Diablo and, so, okay, so I think this one here is an unpaired analysis where I'm comparing pre versus post, right? So this is sort of an analysis where you sort of ignored the fact that these individuals are related. And then the other analysis is where you're using something similar to the differences between these paired observations. Your design scenario might be slightly more complicated because I think you might have like crossover, right, with like multiple conditions, not just pre versus post. You have other things going on as well. And so I think there is like another function in mixomics for like multi-factor classification that you could look into. It's called, I think, multi-level. I don't know if there's a multi-level mixomics.
Yeah, there is a multi-level for SPLSDA.
Yeah, so you could look into this. I know they have they might have a similar study design to what you are trying to model, but even in this design, they keep the test and the validation, the train and the test sets separate when they're doing the cross validation. Yeah, I could dig up my scripts that I had run when I had generated this if you want. I haven't looked at this before, so this is see, they're not even computing cross validation here, so that's something that we'll have to see. But that's a really good point, right? Because obviously you don't want data leakage to occur between your train and test. So that's a very like it's a scenario where you have to be very careful of how you create your models and apply them into the test cohort. I'll get maybe reach out to me after the workshop and I'll point you to some tangible resources. Any other questions?
I do have another one, sorry. It's about the clarification you made, the point you stressed out about when you first identified the 50 features, and then you are clarifying, emphasizing that the error rate was known for those features and that we had to use the predict function, I think, to kind of work with the training, cross-validate with the training data. Could you just go to that slide?
This
There
one?
was
Or is it the p-values, the one, this one?
no, I think it was the one that where you were emphasizing that we need to use towards the end. Yeah, the previous one, I think before that. Yeah, this one.
Yeah.
Okay.
I hope I can knit this properly because this I don't know why, but my R markdown is complaining now. So let me just log out of it.
You might have run out of time.
Oh, no worries. I can look at the slides and see if it all makes sense after. Thanks.
Oh, it's running. We still have like 15 minutes, so I'm happy to answer questions. So here it did run again. So here I can go back to that. Oh, does that slide render? Okay, perfect. Yeah. That was my last slide. I won't go into it now, but it's supposed to have rendered before. I think this was the this one, right?
Yes. Yeah, I was trying to understand the kind of between this slide and the next slide, the point you're trying to stress about using the training data. So that was you using the same data that you used to generate what you have here, right?
Yes, yes.
But
So I
applying the
basically
predict function, right?
this one.
So just the concept between this slide and the next slide. So I remember you stressing that the performance that we see from this, the error rate and all that, is not our actual error rate, and we have to kind of do what you do on the next slide, where you use a predict function and kind of do something with the training data.
Yes, yes. Okay, so I'll explain that again. So what we're doing here is we're testing a grid of values to identify the model with a given number of features, okay? But the problem here is that we have looked at the entire data set repeatedly, multiple times. And then we have gotten a model that has the lowest error rate, but there's some like chance that's attributed to this process as well, right? And to give you the example I had given is that suppose if I had randomly chosen molecules, and I could obviously find panels where I even get a lower error rate, right? But that doesn't mean that that is the correct error rate for that model, because I've literally searched the entire space, literally overfit my training data set, right? So this is going back to main goals, like this is where you know you overfit, where we try like every single model that exists, right? So another example that I might have about that I used to had done before was just that, where I was testing essentially like every model that existed. So let me see if I can find it. Okay, so just going back to that slide again, right, where we have I'm just gonna hide this, where we're trying like these various models of different sizes. You could actually take it further and actually look at other types of machine learning algorithms, right? So there's elastic net, there's just like generalized linear models, random forest, SVM. You can look at like pathways. So like an individual pathway might have 5 molecules. You make a model out of that, right? Now you can literally start testing all of these like millions of models, right? And you're gonna get models that have some AUC that are like very poor, and other models that have a perfect AUC, right? But that doesn't mean that this model up here is going to perform perfectly in a unseen data set, right? So this process of like trying to like tune your optimal process can lead to overfitting as well, right? So that's why we need to actually repeat this process in a cross-validation manner to estimate the correct performance. And that's what's happening on the second slide, is where I've actually put that process in here and repeated it multiple times in cross-validation framework, and then gotten the correct accuracy or error rate. I need to, we can actually fix that there. It's like 13% error rate. So it's slightly higher than the 10% error rate on the previous slide, right? So that's the nuance. Is that clear? I know it's kind of hard to wrap your head around the first instance you see this, but ultimately the main takeaway is whatever you use to create your final model needs to be repeated in your cross-validation fold to get the performance of that final model that you created.
Thanks.
Okay, any other questions? All right, cool. Well, thank you all for attending, and have a great weekend.